Welcome back. Today we're going to do a little bit of a pivot from the recent material, and we're going to dive into the world of electrophysiology. So we've been applying mostly mechanical thinking, certainly some chemical thinking, and now we're going to get into a little bit of electrical thinking about how the elements of the cardiovascular system function, how they're regulated. And we'll follow a similar path we have in the past, starting with structure, getting into function, looking for relationships between the two, and eventually getting to the point of talking about regulation. And in this case, for this particular lecture, we're really going to start into a series of three shorter ones that will cover three different aspects of the cellular domain. So this is cellular electrophysiology, which starts, of course, with membranes and then builds up to whole cells. And we'll live in that sort of space for the next few lectures. Now, the reason I broke it into three pieces is I really see this domain being described with three sets of approaches, three different perspectives. One is the theory, one is the mechanisms, the structures leading to functions as we understand them. So this is sort of the background biophysics of membranes and cells. The next part is experimental, where I think as engineers we do need to be at least somewhat informed as to what sort of experimental methods there are, how they might work, and maybe even where we could find some opportunities to improve things. This is where engineers can come into the picture and have come into the picture over the 50 or 80 years or so that we've really had this field. The third arm is simulation, and that is relevant because that will lead directly into a homework assignment that I'd like for you to do, in which you get to explore some of these behaviors using a computer simulation. So we're going to use a computer to do modeling. This shouldn't be totally new, I hope, for most of you, and a lot of this material should also be, in part at least, review. So I'll go through it fairly quickly in the beginning and refer you to other background materials. And there's many sources of all of this that you can find. And of course, there are references in the materials on the Canvas page. First of all, of course, structure. There are a number of different views of structure, and I'll show you a few of them here. There are lots more great images and explanations you can find in textbooks and on the internet. But the core idea here is that we have a membrane made up of bilipid layers, so two layers of lipids with the hydrophobic tails facing each other and the hydrophilic heads facing out being on the one hand inside the cell, the other hand outside the cell, so intracellular, extracellular domains. We've talked about those a little bit. And this should really be a review of very basic cellular biology, and I can refer you to other materials for that. Embedded in that membrane is a lot of stuff, and people spend their whole lives studying different types of materials, usually proteins, embedded in or living on the surface of these cell membranes. So there's lots here we could talk about. We're going to zoom right in and focus on integrated proteins that form channels, ion channels. So that's really the working mechanism, the real workhorse that we're going to spend most of the time describing and eventually quantifying and modeling. Of course, with a little bit of additional information perhaps about pumps, which we've talked about again before, and they clearly play a very important role in the membrane and cellular behavior in this electrical space. The details, like I say, I'll refer you to some of the figures in the slides and other background materials. There's a nice micrograph to give you a sense of the size scale that we're thinking about here. You see that length bar is 100 nanometers, and some little tiny part of that is the membrane bilayer. You can see this nice image here showing the exterior and the interior aspects, or extracellular, intracellular spaces, and this bilipid layer that's just a few nanometers thick. So we're down to a pretty small scale here, and within that membrane, of course, we have the proteins that we're going to describe, and as if they were big and huge and we could poke at them, but in reality they were very challenging to determine the behavior of. And a lot of this part of the story was really gleaned from very indirect measurements, macroscopic measurements that we'll come to in a later video, that required true brilliance to interpret in a way that describes this underlying structure, which at the time was completely inaccessible to the scientists of that era. And so they didn't have a picture like this, an electron micrograph like this, to base their ideas on, and instead had to really interpret them. I hate to say extrapolate, interpret them from the macroscopic results of the measurements that they actually could make. Now here's a zoomed in or a little profile of some of these proteins. This is just to give you the sense of the complex folded shape that these proteins have. As we know, proteins take on their shape as a function of many things. A lot of the electrostatic interactions determine how their shape behaves. This question of how proteins fold has really been a huge challenge in science, and we're only now getting our heads around it, our hands, our models, to really get to the point where we can predict behaviors of proteins and predict how they're going to fold. But here you see some features of these proteins, and that's really the ion channels that we're going to focus on. They're special cases of these folded proteins. Now the cell, of course, has many compartments. The membrane divides two spaces that can have very different compositions. So they have very different electrolyte concentrations, ion concentrations, very different materials. The membrane separates. Its prime focus, arguably, is to selectively allow certain things to cross, but in general to provide a barrier. So we expect quite different behaviors on either side of the cells. Now there are a number of transport mechanisms that you've hopefully heard of before. This is an overview slide that describes, on the one hand, the passive transport. So by passive, of course, we mean that no energy is directly involved, but also the active transport, in which there are ion pumps, if you will, that will move materials against their concentration gradient. Or sometimes there are indirect mechanisms. There are sort of secondary transport mechanisms that do involve some energy, but in an indirect second-hand way. And again, that's a topic you've hopefully heard something about. Finally, we do have diffusion, our old friend diffusion. That can occur even at rest, even without anything happening in the way of either energy expenditure or a protein channel opening. Certain pores exist in the membrane. Certain materials will cross the membrane quite easily, even when there's not a pore. So there's a whole story here behind all the various ways that things can move across the membrane. And we're not going to get into too much of that at all. Again, focusing on the membrane channels, the ion channels, and a little bit indirectly the pumps. Now, let's jump into the biophysics. So let's get into the structure-function relationships that we're going to talk about or think about here. Before we get into the specifics, though, very brief reminder that we have electricity. So resistance is a key concept here. Here you have an image of serial resistance. We can have parallel resistances. This is also a domain where we'll flip pretty liberally between resistance and conductance. Conductance is a perfectly sensible term. There's nothing secondary about it. We tend not to use it in some domains, but in this domain of biophysics, conductance, the G, uppercase, lowercase, depending on lots of things, will be the symbol that we'll see just as often as we'll see R. And that's the way that biophysicists often think about their behaviors and the way that current flows. But Ohm's law still holds. All the same rules hold as before. Now, another way of capturing the behavior of an element, and it could be very general element, is to capture the relationship between the current and the voltage. So if we apply voltage across this piece of material, I'm being vague here on purpose, but that piece of material, if we apply a known voltage across that piece of material and then measure the resulting current, then we have two numbers that we can, a sequence of pairs of numbers that we can plot on a graph. And so we have what's known as the current voltage relationship, or IV relationship. So in this figure you see on the left-hand side, very schematically, the measurement setup. Again, voltage, a known controllable voltage, so V is the voltmeter. The adjustable battery to the right is the source. That voltage is applied across the object of interest. In this case, I've represented it generically as just a resistance. Could be lots of things. And then in between is an ammeter, symbol A here, with which we measure current. And this basic schematic will come back again and again. This is really how we measure a lot of biophysical quantities, a lot of membrane and cellular quantities. Of course, when we plot these two quantities, one against the other, and we do it with the x-axis being the voltage, the y-axis being the current, then the slope of that line is the 1 over the resistance. So we can also say that the 1 over the resistance is the same as the conductance. So the conductance of an IV curve, when it's drawn in this standard way, will give us the conductance. And that too will be a useful way of characterizing for example, how membrane channels behave. And we'll come back to that very quickly. We have another behavior that we probably haven't thought a lot about, maybe, it depends, but it's this idea that we have an asymmetry around the behavior, the relationship between current and voltage. Here we have, on the left, again, schematic diagram of not just a resistor, but a resistor and a diode in series. Now that will have, obviously, a very different behavior from the resistor alone. And you all know what diodes are. They open, they're like gates. When the voltage is applied in one direction, the gate opens. When it's applied in another direction, the gate closes. That behavior, that directional sensitivity, that we call rectification. That term comes up in a number of settings, but in the biophysical context, we'll see that there actually are ion channels that act in a rectifying way, that preferentially let current flow through the membrane in one direction over another direction. Well, the other direction. There are only two directions here, of course. And so what we now have in a situation like this is an IV curve that is no longer a straight line. The relationship between current and voltage is no longer constant. Instead, as the voltage changes, the relative change in current also changes. There's a lot of changes at once. So one way to capture that is to say, well, what about the slope? The slope is, again, the rate of change of current with respect to voltage. That slope also changes with voltage. And that's a key characteristic generically of a nonlinear relationship. Not necessarily rectifying, but nonlinear. But then we have the special case where if the voltage in this case is positive, and with the right end of this curve, then indeed current flows in a nonlinear way as a function of the voltage. And on the left-hand side of the curve, where the voltage is negative, then no current flows. The red line stays on the zero current axis. So that's a nonlinear resistance and a rectifying behavior, both combined into that one figure. And again, this actually captures key features that we'll see in membrane currents. In fact, here's an example. Here's another schematic where I've put a membrane, ion channel, again, sort of pictographically into the circuit, and that's the same standard circuit you've seen before. And now we see that there are a number of current voltage relationships. And what do they all mean? Well, one of them is the total current that flows through that particular channel. And again, channels, as you know, are ion specific, more or less specific. And even within that same type of channel, say generically type of channel, in this case potassium channel, then we can get many different subtypes. And each subtype has its own different, maybe not unique, but certainly different current voltage relationship. And so you see these curves have different shapes. They have different labels across the top. The I total is the total current flowing through all the potassium subtypes. And then you see three different potassium channel subtypes, each with their own different shape of curve. Don't worry about the red and the black lines. That has to do with the experiment from which these figures came. The point is that there are differences among or between different channel types. And this is how we get the behaviors we do when we capture an action potential, for example, from a cell. Now, the underlying physics that drives a lot of what will follow has to do with this image here that you've probably seen before in one form or another, and that is the notion of passive transport through a semi-permeable membrane and the notion of flux. Some ions can move easily across the semi-permeable membrane, some can't. The end result of that is a potential difference when we have ions, because ions carry charge with them. Another way to picture this, just focusing right down on it, when we think about what goes on, here we have the condition with potassium and chloride being dropped into the left-hand side of a beaker. We're calling that the inside, relatively, of the cell, sort of schematically representing that. The outside is on the right-hand side. And we have a semi-permeable membrane that allows potassium ions to flow. Immediately, as soon as we drop the salt, the sodium chloride salt, or excuse me, potassium chloride salt into the left-hand side, we create a gradient of concentration, a chemical gradient, and that causes, if it's possible for ions to flow, those ions to pass through the semi-permeable membrane and start to diffuse down the gradient and spread over from the inside to the outside, as we've labeled the left and right sides of this beaker. That will occur up until some equilibrium is reached, and that equilibrium is not a diffusion-only equilibrium. It's not an equilibrium in which all the potassium ions, let's say, uniformly distribute themselves, because in this case only potassium can flow. Chloride can't flow across that membrane. The potassium flows across and takes charge with it, and that charge creates a voltage difference across the barrier, across the semi-permeable membrane. We see that in the little voltmeter that's sitting at the top of the diagram. And that also produces a larger and larger electrostatic gradient. So there's a charge gradient as well as a concentration gradient. The charge gradient creates electrostatic voltage, and that voltage eventually starts to repel additional ions moving across the semi-permeable membrane until we get an equilibrium. And at the equilibrium, the concentration gradient that exists is balanced by a voltage gradient, and the voltage at which that equilibrium occurs is, not surprisingly, called the equilibrium potential or the equilibrium voltage. What is maybe surprising, if you haven't heard of this before, is that we can actually capture that voltage, and we call it the Nernst potential. And of course, I think it's hopefully clear that that potential has to depend on the concentration difference, because that gradient is driven by the concentration difference. And if we know the concentrations of ions on both sides of the membrane, the Nernst potential tells us, in numerical terms, quantitative terms, what the resulting voltage will be. And that's the Nernst potential, about which you hopefully have heard quite a bit so far. Now, here are a couple of cases where we can say frog muscle, squid axon, later we'll see cardiac muscle, and of course each species, each type of cell has potentially different concentrations of the major ions across their membranes, intracellular versus extracellular, and that creates different Nernst potentials for each one of those ions in each one of those species. And that species dependence is something that also is quite relevant, of course. Our ultimate goal is often to explain how human heart cells work or nerve cells work, and so we need values that are true to that particular species if we want to describe that behavior with any accuracy. But the basic function, basic concept here is robust across species. This is really core biophysics. It has nothing to do with the species at hand. So now let's talk a little bit about a specific case, the resting potential, before we get into action potentials. The resting potential is the potential when the cell is at rest, as the name indicates. So this is when the cell is not stimulated, it truly is in a rest condition, and many cells will stay at that condition forever as long as there's enough energy around to maintain them. So this is picture this as a stable condition. The stable condition when the membrane or when the cell has more than one ion in it is a little bit different from just the Nernst potential. So the beaker case, it was only one ion really of interest, that was potassium. And so the resting potential of that beaker, if you will, would be the Nernst potential for potassium for those concentrations that we're given. And that's not the case, of course, when you have multiple ions. Each ion will contribute something to the resting potential, and so the resting potential will be an amalgam, an integration of some sort of all of those individual Nernst potentials for each of those ions. And the resting potential almost never sits at the value of any particular ion. As we'll see, it gets quite close to, in this case, potassium, but it's not exactly the same as potassium. So here we have visually a form of diagram we'll see again and again. It's one I think captures the behavior well. What you see is, is a dashed line. It's the cell, sort of a blob that looks totally like a cell, of course. And then to the left, you see the measurement of the membrane voltage. We'll come back to the details of this, but the point is that we're measuring inside the cell compared to outside the cell, and that's membrane voltage. That's also the convention, that it's always relative to the outside. Now, when we have concentration differences on the left here, as we see with potassium, a lot of potassium is inside the cell, so a higher concentration of potassium in the cell than outside the cell, then that creates a chemical gradient. That would tend to push the potassium from inside to outside the cell, hence the blue arrow that goes from inside to outside. Now, as I've said before, as soon as that positive charge starts to leave the cell, it leaves a negative charge behind, and that negative charge, you see with the little negative signs here, means that potassium is going to be pulled back into the intracellular space. So it's going to be repelled or pushed out because of concentration gradient and pulled back because of electrostatics. Those two forces are always at work. And the difference between them, if there is a difference, is a net gradient and creates a net flow of these ions when the membrane is at a certain value. Now, the electrostatic value that you see in this diagram is actually the membrane voltage, whatever that might be. We're looking at it under resting conditions. So let's assume for the moment that's the electrostatic potential at rest. So the resting potential is set at that point. That's a fixed value. It's determined by, as I say, what other ions might be available. They're all integrated into that value. And what we see for potassium is a net difference. So the electrostatic potential here is not equal to the Nernst potential. That's determined by the concentrations. That's essentially the length of that chemical arrow, so to speak. So we have electrostatic arrow, that's the actual membrane voltage, and then we have an electrochemical gradient that turns into potential that's determined by the Nernst potential. Those are the two things that are at balance here, or not quite at balance here, as we'll see. Now on the right-hand side, see the same scenario, but for sodium. And of course, sodium lives in a different configuration in which there are much higher concentration outside the cell than inside the cell. So in this case, the concentration gradient is going into the cell. And that will tend, if nothing else were present, to drive sodium into the cell. Of course, that sodium will also be drawn into the cell because the cell is at a certain membrane voltage. That electrostatic potential is the same. That's the resting potential of the cell. So that potential, being negative inside, will tend to pull sodium into the cell. The concentration gradient will also tend to push or pull sodium ions into the cell. So both arrows point in the same direction, and the net gradient is quite enormous. It's bigger than the individual ones. They add up quite simply and algebraically to provide the net gradient. Now, what that tells us is there's a huge potential for that current to flow. And we know from Ohm's law that current will only flow when we have enough conductance. And so if the conductance is low or the resistance is high, same thing. If the conductance is low, then even though we have a huge potential for that flux, the flux stays very, very small. And it's that balance of different ions and their conductances or permeabilities, different words are used for this same concept, that weighted balance of those is what actually determines the resting potential. And there's an equation for that too. So this is the equation. It looks a little complicated, but it's really doing nothing more than adding each of the contributions from any ions that are relevant to that particular cell. And it's keeping track of the concentrations of those outside the cell and inside the cell. That's the subscript in and out. M and A are the positively charged and negatively charged ions, respectively. And the P's are their relative permeabilities or conductances. Again, it's the same concept. So if we add up all of those terms and we assign values to each one of those terms, then we'll come up with an estimate of the resting potential for that particular cell. So again, this sum over N or M, becomes much smaller. In reality, most of the time we're not dealing with that many different currents, but if we want to be accurate, every current we have to know its relative permeability and the concentration of the related ion. And as we'll see, certain ions, actually we've seen already, certain ions, in that case potassium, can have multiple different channels that all carry potassium. So the potassium concentration gradient is the same for all of those, but they're different in their permeabilities. And so each can contribute a different amount, weighted amount, to the overall voltage that makes up the resting potential. And this equation is called the Goldman-Hodgkin-Katz equation, or the Goldman equation, we'll sometimes see it as. And this is well known, well proven. Here's an example of what we get when we plot the membrane potential as a function of the external potassium concentration. So now we're manipulating the conditions. So you can think we're manipulating the concentration gradient. That's going to change the Nernst potential. It's going to change that component of the resting potential. And we can see here, as that external potassium concentration goes up, then we get a resting potential, or a potential at that concentration. It is sort of a resting potential, but at a different concentration, that falls very nicely along the Nernst equation line. And we see that only at lower extracellular potassium concentrations does the actual membrane potential, the resting potential, follow the Goldman equation line. This is where the relative contribution of sodium is enough that it shifts us away from the Nernst potential for potassium and drives the curve, so to speak, to the left. So on the right-hand side, that right-hand straight line equation, this is a log plot, so it's a straight line. And so that straight line plot, that's the Nernst potential for potassium. So we see at high potassium, external potassium concentrations, then the resting potential agrees quite closely with the Nernst potential for potassium. Sodium doesn't play a big role. But at the lower concentrations, which are the concentrations that are physiologic, right? The external potassium concentration is relatively low. It's in the order of a few millimolars. So it's in that region where the Goldman equation will provide a more accurate estimate, and that's what this plot shows. The circles are actual measured values. So this is how we sort of play this game. This is the thought process behind thinking about how the biophysics works. Resting potential is fine, good to know what a cell does when it sits at rest, but we really have to know about action potentials because that's where, the action happens. That's where things happen, that's where things are regulated, controlled, all that good stuff. So before we get specifically into the action potential, there's a concept that's also important to keep in mind because it helps us understand how action potentials work. It actually applies when the membrane is at rest or the cell is at rest also. Because I said to you before that we don't have, for example, for potassium, as you see in this figure, we don't have perfect balance between the electrostatic voltage, the resting voltage, the minus 80 in this particular case, and the electrochemical voltage, the voltage that comes about because the concentration gradient, otherwise known as the Nernst potential, for potassium. And here we have a value of minus 94. Again, those numbers come based on the concentrations for a particular species. It's not so important here that you know which one. But we have a resting potential of minus 80. We have a Nernst potential of minus 94. There's a difference between those two. And that difference, we can do the math, it's not hard, that difference is 14 millivolts. And we describe that difference as the driving force. That is the potential for, in this case, potassium to flow. It's the size of the voltage difference. It's that term in the Ohm's law that we have, typically in the numerator that says that's the voltage difference. And the flux is going to be that difference times the conductance. So it's not the entire electrostatic voltage or even the Nernst potential for potassium that determines the push or the gradient or the potential for ions to flow. It is the difference between the electrostatic at the moment and the chemical Nernst potential at the moment. And as you can see here, it's a much smaller voltage, and that's not unusual. Now, of course, the driving force for other ions will be different. The driving force for sodium is huge. And, because if you do, again, this simple math, you'll see that both these terms add up. And so we get a 80 millivolt plus the Nernst potential for sodium. And so we get an enormous potential for flow. But without enough conductance, no flux will happen, no ions will actually flow. So that's called the driving force. And the driving force is what we feed into our equations. So here we see the flux equation I was just telling you, reminding you about. So the flux equation where we have current flow equals the difference between the membrane potential and this equilibrium potential or Nernst potential for the particular ion in question, divided by the resistance for that particular ion channel type. This could also be this driving force, the Vm minus Veq, multiplied by the conductance. Same equation exactly, of course. We can also manipulate this equation in kind of an interesting way because we're going to see for action potentials, the real behavior description comes in how we can predict the change in the resistance or conductance. And if we write the expression this way, putting resistance, let's say, on the left, and the voltage, the driving force in the numerator and the current below it, if we knew the equilibrium voltage, which is just driven entirely by the Nernst equation, so if we know concentration, we know the value of the equilibrium potential. The membrane voltage we can actually apply that voltage across the membrane. And then we can measure the associated current. So this is a little bit like those IV curves that you saw before. In this case, it will deliver to us the value of the resistance. And we can do things like change the membrane voltage and see how the resistance changes and learn how resistance depends on the voltage that is applied across the membrane. We can also, if we're clever, do this in a way in which we can capture the time dependence, because we will see that resistance, conductance changes not just with the membrane voltage, it also changes as a function of time. So it's a fairly complex story that we'll spend a lot of time talking about and even simulating. So this equation really gives us a powerful tool to both think about and eventually measure in a way that delivers these values of resistance or conductance in their sort of full subtlety, how they depend on the voltage, how they depend on time. The cardiac action potential is different from the nerve potential, and we'll show you a little bit more of this in the measurement section, but conceptually we should know right away that cardiac action potentials are much longer than a nerve action potential. Nerve action potentials are a few milliseconds. An action potential in the heart, it varies for sure from different tissues, from tissue to tissue, or cell type to cell type within the heart, but it's in the range of 300, let's say, to 350 milliseconds, somewhat broadly like that. It can be longer and shorter, but it's a ballpark sort of number to keep in your head. So it's much longer. That's the main point here. And we can associate each different phase of the action potential with different ion channels. This is another core way of thinking about cellular electrophysiology in any cell type, is that we get voltages across the membrane that are a function of the underlying currents that flow. And that, again, Ohm's law would tell you that. So in this little diagram, you see that we have a generically shaped action potential, a cardiac action potential. You see resting potential here is around minus 80 millivolts, a little more positive in this case. The peak potential goes a little bit above zero. And we see that at each phase of the action potential, different currents are active, which is another way of saying that for each point in this action potential, the underlying ion channels are open for that particular ion type or that particular current. So in the beginning, we have the sodium channels open, and the upstroke, we call it, that rapidly rising phase of the action potential, that's carried mostly by sodium currents because the sodium channels are open. Then the potassium channels turn on and the calcium channels turn on. And so they start to act as we get a little bit later into the action potential. And they last throughout the rest of the action potential, but not at constant values. None of these are constants. Even the sodium current is not a constant. It turns on and it turns off with some shape. The potassium turns on and then it turns off with its own shape. And as we said before, multiple types of potassium channels will have different shapes. And similarly, the calcium current will open and turn off with its own shape. So the complexity then lies in these shapes of the actual currents, and these shapes of the currents are going to be a function of the conductances and how they change with time and voltage. Here's another version of that same information. Here we see it's a nicely done figure, shows the different phases of the action potential. These are also standardized, phase 0, 1, 2, 3, 4. And during each one of these phases, you can see below which of the currents are active, which of the channels are open, and that helps us understand the behavior of an action potential. The key thing to remember, though, is even though it's an action potential, if we take any point in time, the same ideas really hold as held for the resting potential. It's just the conductances have changed. So you can picture a Goldman equation that you could write down, and if you could track the changes in the conductance, you could predict what the membrane voltage will be. And that's the mode that we'll use in the simulations that we'll see later on to try and capture this behavior mathematically and then eventually with a computer. Here's another diagram that shows, again, the action potential in the middle, and above it, a set of red lines, and below a set of blue lines. The ones above are called depolarizing. So those are the currents that contribute to the depolarization. Depolarization is what happens when we take the cell from its polarized state, which is what it is at rest, and depolarize it. So polarized means that the voltage difference is large, meaning inside and outside. It's negative at rest for all the cells we care about. And depolarizing it would drive that potential from the initial negative value up to progressively more positive. So anything that depolarizes will cause the membrane voltage to become more positive, and anything that repolarizes, any current that repolarizes, will tend to restore that polarized voltage, will tend to pull the voltage across the membrane back into the negative domain. And the labels shouldn't surprise us. The sodium current is a depolarizing. It caused the upstroke of the action potential. The calcium currents are also depolarizing currents. There's also a calcium-sodium exchanger. It's another more subtle feature. All these are depolarizing features. The blue are the potassium channels typically. They are the repolarizing ones. They are the currents that flow that tend to restore the cell back to its resting potential. And you can see the different tracings, those different shapes I just talked about, captured schematically, at least in this particular diagram. Here's another diagram that really also tries to capture this idea that here we see a membrane voltage again during action potential, 3 different time points, T1, T2, T3, that I've very loosely highlighted here. And in each one of them, you can see a little diagram just like we had when we pictured the cell at rest. And we have an electrostatic potential, which, depending on where we are in the shape of the action potential, will vary. So those mustard-colored arrows will vary. And we will see their impact relative to the local gradients that exist. So these are the concentration gradients and their respective Nernst potentials at each one of those time points. So we can see the arrows are moving back and forth. This would probably make a great animation. The arrows move back and forth depending on both the electrostatic potential at that time and the concentration gradients, and the resulting amplitude of the currents that flow will be a function of the conductivity, and will also be a function of the charge that's on the ion. So you'll notice that calcium has twice the charge on it that a sodium or potassium ion has. That's the little 2 in the superscript. We all know what that means. So some of these arrows will look artificially longer because they're playing out with twice the impact because it's a calcium ion. So many ways we can see this idea and picture this notion of driving force and use that to predict behaviors. Now, what do we know mechanistically about the behavior of, for example, the sodium channel? I've already mentioned it opens, it closes, what the heck's going on here? How do we picture this? This is part of the insight, the intuition that came from these original experiments, the Hodgkin-Huxley experiments that won them the Nobel Prize. These are experiments that took place mostly in the late 40s and 50s. And they hypothesized, without having much more than macroscopic measurements, the idea that there are 2 openings or 2 gates that control the pore of the sodium channel. And here you see the scenario. So on the left we see a sodium channel at rest, and you see a little ball hanging off the bottom, and then you see a couple of indentations halfway up the channel. Those indentations halfway up the channel mean that the channel is closed. So in this configuration, that little sodium ion, that little orange sphere, will not be able to get through the channel because the channel is closed. Now, if we activate, if we stimulate, if we depolarize through some means the cell and push that membrane potential up to the threshold, that will cause those indentation gates that schematically represented there to open. And as soon as that opens, the sodium will flow through because there's an enormous potential. Remember, there's an enormous driving force that will push sodium through that opening if the opening allows it, if it actually is low enough resistance or high enough conductance to flow through. So that upper channel opens, sodium will flow through. That's what happens during the early part of the action potential and causes a large spike in the sodium current. Now, it turns out that as a function of time, again, the reasons for this are complicated and more than we need to know, but the bottom line is that as a function of time, that gate at the bottom will then close. It's like a ball and chain is sort of the shape of the protein, so that that ball of protein swings up through a conformal change and blocks the output or the bottom of that channel and stops the channel flowing again. That's called inactivation. So we have activation during depolarization, we have inactivation shortly after. This plays out in a few milliseconds, and then the sodium current will stop. And then it will stay in that closed behavior for some period of time, and then eventually that gate that was originally closed will close again, the one that opened briefly, that activation gate, it will close again, and then the cell for a time is doubly blocked. So truly nothing will get through. Both of the individual gates are closed, and then over time, and there's a recovery process that has its own interesting subtleties, but there's a recovery process that occurs that allows us to go back to the state where that ball of protein falls back out, and we only have the one aspect of the channel, one gate of the channel closed, and we go back to the resting case. And this cycle is what happens with every action potential in which there's a sodium channel. This is true of nervous system membranes and cells, nerve cells, as for virtually all other cells in which there are sodium channels. We think this is somewhat preserved across different tissue types. So that's how sodium channels work. Each one of the different channel types has some similar story. Potassium has its story, calcium has its story. This is one very schematic situation to sort of describe all this, in which we have, in this case, the cell membrane at rest. And you see the sodium upper gate is closed. So sodium has two gates. There's two little arrows there. The upper gate is closed. That's that indentation in the previous image. And the lower one is open initially. The same we think is true of calcium. Calcium seems to have the same behavior of a gate that's initially closed and one that's open when the membrane is at rest. The potassium channel is generally closed, a little bit open, maybe has a little bit more conductivity even at rest than the sodium or calcium. But it's only relatively more conductive. It's not nearly as conductive as when either the sodium or calcium channel is open. And then I've also included here with these circles the various pumps and exchangers that are involved. Now these also are important for the longer term maintenance of the concentration gradients that I just sort of told you existed. I said potassium is in higher concentration inside the cell, and sodium is in higher concentration outside the cell. The reason that is because these pumps establish and maintain that balance, and they take energy to do it. So these are active pumps, and you should have heard of them before. There's another sodium-calcium exchanger. It's not quite a pump. It's a little different than a pump. That one has an important function in cardiac cells, not in all different electrically active cell types, but certainly in cardiac cells, as does the calcium pump, which simply moves calcium with energy consumption from the inside to the outside of the cell. So these are all the ion channel mechanisms and the pumps that are at work that give us action potentials. And here's another summary of the action potential with the different currents that are responsible for it. Again, we can get more and more detailed depending on how subtle the effects are that we want to change or what we're trying to study as electrophysiologists. So that's it for the basic background. If you can get your head around this notion of driving force and what it means, you're a long way there. It will take a little time to think through. We'll try and reinforce that in some of the discussion sessions. But if you can sort of keep that notion clear in your head, then the rest of the behaviors actually become pretty straightforward, or at least the unknowns are, predictable unknowns. They're things that are hard to measure, or things that are not very stable, or things that are affected by drugs, or mutations of the protein structure. All sorts of things can play a role, but the basic function is hopefully fairly clear. All right, that was the first of three. Stay tuned or come back later, and you can jump into the next video where we'll cover the measurement techniques that engineers have developed and physiologists have used to explain all this, to really get to these nice clean explanations that I've just given you. See you in a few minutes, hopefully.